we go, what the heart rate is going to be. And it does it by forming a cardiac conduction system. Okay? Cardiac conduction system made of myogenic nodal cells. Okay? The cardiac conduction system is a pathway of these myonodal cells. The pathway starts at the top wall of the right atrium. It sends signals from one myogenic nodal cell to the next, down to the fibrous septum, and then down into the ventricle. This signal, which is essentially instructions of when to contract, how fast to contract every minute, is spread across the atrium and across the ventricles so that the heart will contract pretty much at the same rate. All the muscle fibers contract at the same rate, the same number of times every minute. Okay, so that's very important. That's our cardiac conduction system. Now let me show you the whole conduction system, and then we'll go through it piece by piece. Yes, I know. I just want to... It's all right. I'll go back. I'm going to go back. All right. This is essentially the cardiac conduction system that you see here in red. Okay. We have our atria up here, our ventricles down here. Here's the fibrous septum. Now, normally the syncytium of muscle fibers of the atria cannot communicate with the syncytium of the ventricular muscle fibers because of that fibrous septum. So we have these myonodal cells, and they form structures. This first red dot right here is called the SA node, the sinoatrial node. That's the first structure. That's where the signal will originate. The signal will then be passed to the next structure of myonodal cells called the AV node. That actually AV node should be sitting right over here. And that AV node stands for atrioventricular node because it sits between the atria and the ventricles. And this augments the signal and then passes it through a line, this line right here, which crosses the fibrous septum. This is called the bundle of His. And that bundle of His then branches into two bundles, which go down either side of the interventricular septum. These are the right and left bundle branches. And coming off the bundle branches, going into the myocardium of the ventricles, are these fibers. They are called Purkinje fibers. And all of these structures are going to carry an electrical signal that tells all of the cardiac muscle fibers when to contract. Okay?
What if there's damage to the AV node?
Oh yeah, if there's damage to the AV node, then you won't have a signal going down into your ventricles, and that's the problem. If you damage, if you lose your SA node, your atria can still get a signal from the AV node. And you lose your AV node, you don't get coordination of contractions between the two bundles. You end up with problems. And then this will end up with an atrial tachycardia. Your atria won't be getting a signal, so what will happen? It will be at its own syncytium, 120 beats per minute. You had a question?
So I'm not making only one node, or there are others somewhere.
Well, there's two, the SA node and the AV node. Those two nodes, and then there's the bundle of His, the right and left bundle branches, and then the Purkinje fibers. Okay? So that is the pathway, that's all of the structures are part of this conduction system, the cardiac conduction system. Okay? So we start, okay? All right, so let's look at the SA node to start with. Myonodal cells are collected in the top part of this chamber. And this SA node can receive impulses from, for example, the brain, telling it to speed up or slow down. However, it makes its own signal. It's able to say, okay, our cardiac cycle will occur 70 times per minute. We will have a heart rate of 70 beats per minute. And 70 times every minute, the SA node will send out an electrical impulse. That electrical impulse will be sent from the SA node to the AV node, which sits right about here. The impulse also travels out across the muscle cells or muscle fibers of the atria. So essentially, whatever the SA node defines in terms of how many beats per minute, the impulse will travel across the atria. Wherever that impulse, it's an electrical current, wherever the electrical current travels across, it causes the muscle fibers to contract. So as the current spreads across these chambers, the chambers start to contract, and that is atrial systole, okay? Then that signal also is sent to the AV node, okay? From the AV node, the signal will then have to cross the fiber septum, and it does it, can I go to the next one?
Yeah.
Okay. It does it by crossing over the bundle of his, this one right here. And we can see here, AV node picks up the impulse, transfers it to the bundle of his. Okay? Right there. Once it crosses into the ventricles, it travels down the right and left bundle branches and into these Purkinje fibers. Remember, this is an electrical current. Once the current goes into the myocardium of the ventricles now contract, and we have ventricular systole. This series or sequence of events occurs however many times per minute that you have your heart rate is. Okay? And it should take a particular period of time for the signal to get from the AV node, SA node to the AV node, or down the bundle branches. You can see there are, 0.03 seconds from the SA node to the AV node, a very short period of time. We're not so much interested in that right now. If you were going to be reading electrocardiograms, you'd be interested in it. But what we're interested in is knowing this pathway. Know the pathway of the conduction system. SA node to the AV node, to the bundle of His, to the right and left bundle branches, to the Purkinje fibers, okay, and into the myocardium, all right? So that's important to know. Know that pathway, all right? Now, here it is again. Again, showing the pathway of the electrical event. And what is it that we're interested in? That even though we can't see, when we hook somebody up to an electrocardiogram, and it shouldn't be EKG, that's not appropriate, electrocardiogram is an ECG machine. Yep. When somebody is hooked up to an electrocardiogram, that machine is reading the electrical impulse as it travels from the SA node to the AV node, to the bundle of His, to the right and left bundle branches, and into the ventricular myocardium through the Purkinje fibers. That's what the electrocardiogram is reading. It's not telling you when the atria are contracting or when the ventricles are contracting. It's not telling you that. It's just reading this electrical current. And we're going to look at an electrocardiogram, and we're going to put each of the events of this electrical current traveling across the myocardium into perspective. Okay. All right. Da-da-da-da. This is just showing, the small cells that make up the SA node. The SA node is actually found a tiny collection of cells in the superior or the roof of the aorta. Okay, it initiates the heartbeat. It generates its own electrical signal and initiates the heartbeat. Okay, initiates that heartbeat. Okay, the signal then goes to the AV node, and this is and I wish we could have been able to do this. It's very hard to dissect out the cardiac conduction system from a preserved heart in the lab, but it would have been nice to try. But from the SA node to the AV node, and then from the AV node, we will go down bundle branches. We'll have here's a right bundle branch and a left bundle branch. Where's our left bundle branch? I don't see it. And then Purkinje fibers will be actually under these trabeculi. Okay, so again, this conduction system is very important. Now, let me just ask you something. Say somebody has a blocked artery, a blocked branch of one of their coronary arteries. and they end up with what they call myocardial infarction, a heart attack. When an artery is bringing blood to this myocardial wall, this muscle, and the artery is blocked, blood doesn't get to the muscle. And what happens? The muscle will die. It'll die. And when it dies, a lot of this muscle, particularly the ventricular muscle, imagine two things. Where's our aortic valve? Let me go back to the other one. I'll show you this one. Here. I don't know if you can see this, but this actually is part of the tricuspid valve right here. Okay? So imagine, here's our tricuspid valve, here's our chordae tendineae, and there's the papillary muscle it's attached to. Imagine an artery bringing blood to this tissue right here was blocked, and this tissue right here died because the artery was blocked. If this tissue dies, look what's attached to the inside, the papillary muscle. That papillary muscle will also be affected. It might also die. If that papillary muscle is affected, then the chordae tendineae attached to it may also be affected, and it can actually result in damage to the tricuspid valve. So a heart attack can result in a murmur, a valve murmur, because of the damage to a particular part of the vessel wall. But not only that, not only can you get heart murmurs because of valve damage, you can also have parts of the vessel wall or the myocardial wall damaged from a blocked artery, and then what you have is you have some of these Purkinje fibers die off. Or you could even have a blocked vessel that carries blood to the atria, to the right atria, and it can cause damage to the AV or even the SA node, and that can disrupt the heart rate. So myocardial infarctions, depending upon what vessel is blocked and what tissue of the heart has died, different structures can be affected. Okay. All right. So let's look at this electrocardiogram really quickly. This is a basic electrocardiogram rhythm strip. What we're looking at is one heartbeat, just one heartbeat. Typically you do an electrocardiogram, you get this long strip, it's just a few seconds, and it has several heartbeats on it. This is just one heartbeat. And what it is a recording of the electrical impulse that has started at the SA node, gone to the AV node, down the bundle of His, then the bundle branches, and into the Purkinje fibers in the ventricle. And that's what we're reading here with these bumps and lines and complexes. And we're going to have to associate in our electrocardiogram every bump with the electrical event that occurs in the heart. And we're going to go through each of them, okay? And let me just show you here that in our electrocardiogram paper, I'm not going to teach you how to do. If you guys want a lecture for reading electrocardiograms, they have great lectures. I'll be happy to give them to you. Just let me know. But we're not going to be covering that here in our class. But we can see the paper itself is divided into little squares, and that's to help you figure out how much time has gone by for each event to occur. And as you can see, 5 little squares equals about 0.2 seconds. So